All right, I think we are ready. So, good morning, good afternoon, everyone. And I'm today's host, Maggie Lau. And today we have a treat because we have two speakers instead of one. And we have Anna Wang and Albert Fahrenbach. And so they are both the members of the Australian Centre for Astrobiology, and they're also affiliated to the School of Chemistry at the University of New South Wales in Sydney. So without taking too much time from them, today we'll learn about RNA precursors and also primitive members. So Albert can take it away.
All right, thank you, Maggie, for that introduction. So hello, everybody. For those of you that don't know me, I think it's probably most of you, I'm very happy to introduce myself and make your acquaintance. So I'm a lecturer at University of New South Wales, and I'm also a chemist in the original life field. And today I want to talk to you about a paper that I recently published called Continuous Chemical Evolution Talk. Is just kind of give you a feel for the type of stuff that I'm interested in, and tell you a little bit of a story about some chemistry that's not too heavy on the detail. So my internet can be kind of laggy, so I'm just going to turn off the video just to keep things running more smooth. Okay. So I hope everybody can still hear me. And here we go. Oh, by the way, please feel free to check out my website. And you can kind of follow some of the stuff I do at if you follow this Twitter label as well. Okay. So just a little bit about myself. Originally, I'm from the US, if you can't tell from my accent. I was born in Indiana. I did my undergrad there, and then I moved to Chicago, or close to it. I did my PhD at Northwestern with Fraser Stoddart, and he's a big name in molecular machines, in artificial molecular machines. So I thought to myself after I graduated, wow, artificial molecular machines shares a lot in common, conceptually, with the original life research field. You know, we're both interested in things like non-equilibrium thermodynamics and template-directed synthesis and how to make molecules do work. So after that, I wanted to get into the original life research community, and I applied for a position with Jack Shostak's lab. He's also kind of famous guy in this prebiotic chemistry scene. And at the same time, I was lucky enough to have this affiliation with ELSI. And I was going back and forth between Boston and Tokyo, spending about equal amount of time at each place. And that's where I got to know ELSI, and it's such a wonderful place over there. And I stayed at ELSI for about another year after that as just an independent researcher. And then this job opened up at University of New South Wales, where I could start a group and do chemistry in original life chemistry, specifically for that, for prebiotic studies. So I moved to Sydney in 2018, and you know, Sydney's just, it's a great city. Here's a view of the beach close to where the university is. This is the building that Anna and I work in. It's brand new. So Anna and I also share labs. So this is a picture when the lab was just built, and that was finished, gosh, when was it? I guess close to sometime about last year now. And so this is the other part of the lab. This is like where we keep all our analytical instruments. So we're pretty much decked out to do a lot of synthesis, organic synthesis, and a lot of analysis. So some of the instruments we have are a QTOF LCMS, a GCMS, an RNA synthesizer, an analytical HPLC, prep HPLC, and we even have our own crazy gas lab set up where we can introduce different types of gases, like anything from SO2 to H2S, NO, you name it, we pretty much got it. So the reason I'm telling you all this is if you have some crazy chemistry experiment that you really want to try, well, just come talk to me. Let's try to get it done. Okay, so enough about my background. So in terms of the research that I'm interested in, here it is in a nutshell. So I guess a lot of you have probably seen this picture before. It's such a great picture. So on the left-hand side here, you have the Big Bang, galaxy formation, evolution, and then somewhere in here is the formation of the solar system, accretion of the Earth, and then here's prebiotic Earth. And then something happened a long time ago on the Earth, about 4 billion years ago or so, that caused this switch from chemical evolution, there's everything on the left-hand side here, to biological or Darwinian evolution, everything on the right. And in a nutshell, what I'm really interested in is what happened at that point in time. How did that switch occur? And so to kind of give you a more, I guess, let's say, poetic interpretation of the problem, because I know most people here aren't chemists, I want to introduce this concept of the Library of Babel. So Library of Babel was a short story written by this author, Jorge Luis Borges. And basically, it was a story about this library that contained every possible book that could ever be written. So the Library of Babel contains all possible books that could ever be written in a given alphabet. And it's all possible permutations of letters on a page. And so this library is vast. But almost every single one of these books is just complete nonsense, you know, because it's just like the monkeys, 1,000 monkeys with 1,000 typewriters. You know, if you just let them go at it, most of it is just going to be nonsense. But somewhere in this library is all the works of Shakespeare, every science paper ever written, you know, and even the story of how life got started on Earth must be in this library, right? It has to be. And so something interesting somebody did online, this guy named Jonathan Basile, I have no idea who he is, but he made this website which models the Library of Babel online. So you can go and search through this library. And so here I was browsing through the Library of Babel online one day, and I was just looking through, and I happened to select a book out of the shelf here. And wouldn't you know, this book actually contained the abstract from my talk. Here it is. In case you didn't read it, here's another chance. Of course, that's not what I really did. If I was browsing through this library, I would have never found it. You can actually search the library for text that you want. But, you know, here's page 260 of this particular book. Sorry, the page right before it, 259, just complete nonsense. And then the page after it, just complete nonsense as well. So you have something that seems to have meaning just sandwiched in between nearly infinite array of just nonsense, stuff you can't interpret it. So to me, the Library of Babel kind of is similar to the problem that we face as a prebiotic chemistry community. So is prebiotic chemistry like searching through a vast chemical library of Babel, where every atom is a possible letter, every molecule a possible word, every reaction a possible sentence, every network of reactions a possible paragraph, every geochemistry a possible page, and every exoplanet a possible book, right? And maybe we don't really know at this point, the vast majority of all this is just Babel, right? And so this is kind of a fear that we have, I think, as chemists in the community. How do we really kind of reconcile this huge, vast chemical space, you know, this complexity? And, you know, prebiotic chemists are not the only people that fear this type of complexity. It's really the chemistry community as a whole that I would argue fears this, you know. But prebiotic chemistry has to really begin embracing this complexity if we are to begin sifting through a vast chemical library of Babel. And so the story I'm going to tell you about today for the rest of the talk is a story about complex chemistry. And so this was based on a paper we recently published, so feel free to check that out if you want. But in order to get the complex stuff, we first have to start from some simple conditions. And so this is kind of where I'm coming from in terms of the research I'm interested in. So I'm starting with the idea of a warm little pond, you know. So sometime on the ancient Earth, you could have had some dry land forming, kind of like through these volcano hotspots like the Hawaiian Islands or something like that, in these geyser systems. And so that's, you know, a warm little pond that can dry down and evaporate, something like a tide pool as well. That type of setting, as opposed to something in the ocean or underneath the ocean. And okay, so that's kind of the environment. Well, what about the molecule? Well, in the rest of this talk, I'm going to be talking about HCN, hydrogen cyanide, as the starting material. And so HCN can be produced from spark discharge experiments that model lightning. It's been observed in space and comets in the atmosphere of Titan. It can also be formed through meteorite impact on the Earth, just through that sort of really hot, intense chemistry. And so, you know, a lot of people, not just myself, think this could have been a common starting material. So the other thing I'm going to include in these in my starting conditions for complex chemistry is salt. I don't think anybody has a problem with just sodium chloride. And then we're going to add a pinch of ammonia, a pinch of phosphate, and of course, water. So that's where we're going to start from. What about energy, though? So those were the starting materials. How about energy? Well, here we're going to use radical chemistry. And so what happens when you take chloride and you take water and you just zap it with some energy? So these lightning bolts don't mean literally lightning. They just mean zap it with some type of energy source. When that happens, it's very well known that you can kick off an electron from chlorine, let's say, or chloride, and make chlorine radical, as well as this thing called a solvated electron. A similar thing happens to water, except you make a solvated electron in a OH radical. So these are powerful oxidants, the Cl and the OH, and the solvated electron is a powerful reductant. So we get this redox power in the same reaction all at the same time, which is really good if you want to make different types of organic molecules in different redox states. And so there's plenty of ways to get these species, which I would also argue are pretty fundamental in terms of being a prebiotic reagent. So you can get them from UV light. You can get it from longer wave UV light with transition metal catalysis, and you can also get it from ionizing radiation. And in the talk that, the rest of the talk that I'm going to talk about today is going to focus on gamma rays. And okay, so finally, we have to think about what sort of rules we want to play by when we do these types of prebiotic chemistry experiments. And you know, it's different from traditional. Hello?
Yes,
Hi, we can hear you.
You can hear?
Yes.
Oh, great. Okay. So traditional organic chemistry is a very different thing from prebiotic organic chemistry. So in traditional chemistry, we have all these different purification techniques. So we make something and we can purify it and make it nice and clean. But in prebiotic chemistry, we can't do that. Also in prebiotic chemistry, we have to think about synthesizing molecules in the presence of many other molecules as a consequence of the fact that we can't purify stuff. And likewise, we have to think about optimizing a yield of a complex mixture of molecules instead of just focusing on one sort of target. And you might just summarize this as saying no human intervention, right? We want to minimize the amount of human intervention that we're applying to these prebiotic syntheses. And of course, we can't just choose any sort of starting material that we want. It has to be something earth abundant. Okay, so this is where this idea of a continuous reaction network comes in. So continuous reaction network is defined as one which is minimally manipulated to most accurately simulate a natural environment in which intermediates are not purified, side products are not removed, and new reagents other than the initial starting materials are not added. So it's basically just saying, you know, you just have to let the network evolve and see what happens. You know, what kind of complexity does that lead to? And so therefore the synthesis of target molecules must occur in the presence of whatever side products are also formed as a consequence of these initial conditions. So the basic idea that we're trying to do is this one. Let's just start from some simple organic and inorganic feedstocks. We're going to apply some sort of energy. We're going to do some wet-dry cycles because we're working on dry land. And maybe we can get to all the way to something like RNA, which a lot of people think was an important biomolecule that led to life's origins. And that follows on the heels of the RNA world hypothesis, which I guess most people here probably heard of, so I won't get into that point too much. Okay, so here let's meet some members of the team. So the people most involved in this project are Jim Cleaves, Zach Adam, Ri-Chin Yi, and Jeremy Tran. He's my honor student at UNSW, and these folks are all people associated with ELSI somehow. And really, Ri-Chin's face should be on every single slide because he's the one that actually did a lot of the experiments you're about to see. So anyway, this is a picture of the gamma radiolysis facility at Tokyo Tech. And these are some of the vials that we were irradiating. So gamma rays are actually pretty convenient to work with because they can pass through anything. So all you got to do is just fill up your vial with some the solution you want to test, and then expose it to gamma rays, and then come back and pick up and analyze what you got. So this is what we did. And so the basic idea, again, just to remind you, is that we're going to take some salty water, we're going to blast it with some gamma rays. That's going to make these OH and chlorine radicals, as well as this solvated electron. So this has oxidizing potential, this has reducing potential. We have some cyanide there. We have some phosphate, a little bit of ammonia. And the idea is, well, maybe we can get all the way to something like a ribonucleotide. And the reason we thought this was possible because we knew of this kind of now famous pathway in the literature called the Pounder-Sutherland pathway. And these guys really kind of, I think, revolutionized the field when they showed that you could actually make RNA pretty simply following this pathway, starting from some pretty simple stuff. So don't worry if this looks confusing chemistry. The point is that we know kind of the molecules that we want to generate out of this. And so our first sort of target molecule was this guy in red, this 2-aminoxazoline. And that kind of represents a halfway point to being able to get to something more familiar like the ribonucleotides. So that's what we did. So we did the experiment. We took some of the salty water with our ammonia and phosphate in the beginning. We blasted it with some gamma rays. We saw that we're making some of these important compounds that we saw on the previous slide for making RNA. And then when we would do a wet dry cycle, that would allow us to actually get to this target, this 2-aminoxazoline. And that wet dry cycle had some kind of important function. It helped make the reaction proceed efficiently, mostly by getting rid of the HCN, but I won't go into the details unless somebody wants me to later on. And yeah, so this is about as complicated as the show. This is just a little bit of data for those of you out there who like this sort of chemistry in NMR. So this is an NMR spectrum of this experiment that we did. And it shows after you're done irradiating and drying down the solution, the NMR shows that you can make this 2-aminoxazoline, or sorry, 2-aminoxazole in red, as well as a lot of other kind of fascinating compounds. And if you zoom out of this NMR spectrum, so we were just looking at this zoomed in region, but if you took a look at the whole spectrum, you can see we're making a lot of other stuff at the same time. So this really is a complex mixture. We haven't even identified everything we're making yet. But the point is that the chemistry still works. And so this is where we're at now. So we have this kind of complicated scheme going on. We start up here with just some salty water, we irradiate it, and then we have some cyanide in that solution, and we go through this complicated pathway. And we're at this point right here, this aminoxazole in red. And these dotted lines are the steps we still need to do. So you can see, we're roughly maybe like halfway there. So after a day, you can get halfway to a pyrimidine ribonucleotide, which is not bad. I think over time we'll eventually show that it's not so hard to get to the full ribonucleotide itself. Okay, so that's all from me. I've tried to keep it short and sweet. So the take-home messages are, we should try to embrace complex chemistry as a community. I think if that's the one thing you learn from my talk, is that that's the only way to, I think, make significant progress in this field. And maybe the only way to make sense of this chemical library of Babel, so to speak. Maybe we'll find it's not full of Babel after all. It's actually full of patterns that kind of repeat themselves, and we can really make sense of it. And finally, if you want to collaborate, please reach out to me. I think you probably all have my email, I guess, through the listserv, or you can check out my website and get a hold of me there. All right. Well, with that said, thank you for listening, and just like to thank all the people that participated in this project, and thank UNSW for their support, as well as the LC at Tokyo Tech and ANSTO. Okay, thank you.
Hi, everyone. So what I wanted to do before talking about research was give a quick plug for a conference that we at the Australian Centre for Astrobiology are hosting in September. So it's an easy date to remember. It's September 9th, and it'll run for 2 or 3 days. Please go to our website for further details. So that's aam2020.org, and that stands for the Australasia Astrobiology Meeting. And what we want to do, because this will be hosted online, is of course to engage with our neighbours in the Asia Pacific region. So yeah, we'd love if everyone at ASAP could join us. And yeah, so please remember that website and have a visit. What I want to do is to have the director of the ACA, Martin, introduce a bit about what the ACA is about, and then also the sort of research that he's been doing. Because Albert's a prebiotic chemist, but that is just one tiny piece of what happens at the Australian Centre for Astrobiology. So Martin, would you care to unmute yourself?
Yeah, no, I think I am unmuted. Can you hear me?
Yeah.
Yeah.
Great.
So so the ACA has been around for about 12 years or so, and started off at Macquarie University in Sydney, and then transferred to UNSW. It was led and established by Malcolm Walter, who's an expert on stromatolites, and actually found the oldest stromatolites on Earth back in 1980. And he forged a very strong working relationship with people at NASA at the time when they were doing some of the exploration of Mars. And so that connection led to the ACA becoming officially affiliated with the NASA Astrobiology Institute. And the connection with Mars research remains very strong, based on our understanding of ancient life on Earth, and primarily from the Pilbara region of Northwest Australia. So we run field trips there every year, every 2 years, for groups from all over the world. We conduct research. I've got PhD students there. And the link to what Albert and Anna do is really to try and bring some deep time sort of reality context to this idea of, what was early Earth like, what do you need for prebiotic chemistry? Well, let's look at the ancient rock record. Do we have conditions X, Y, Z that would be favorable for these kinds of setups? So that's where we're really starting to overlap. The ACA also has a group that studies microbiology. We've got Brendan Burns, who studies the microbial makeup and metabolisms that go on within microbialites, so stromatolites, microbial mats, that sort of thing. They're unbelievably complicated communities that do quorum sensing and exchange elements and change their environment, so cause mineral precipitation. Then we've got a group that looks at exoplanets down in the School of Physics. Chris Tinney and his group have got quite a lot of time on some of the very big telescopes to search for exoplanets, and have found quite a few. And so we're engaged across quite a range of interesting kind of research. I mean, astrobiology is as big as you want it to be, because it's really just studying the natural world and then thinking, how can we apply that to other planets and how they work, and whether or not they could have the conditions for life. And one of the things that I've been very interested in, I'm going to end up here, I'm not going to go very much longer, but NASA's mantra for astrobiological research and solar system exploration has been to follow the water. And that really is about when you already have life established. course, all life on Earth needs water to survive, to do its metabolisms. But interestingly, the conditions for prebiotic chemistry, and Albert touched on this a little bit, are actually quite different. So there's a big problem with having too much water. And so we're now very interested in this developing idea, and we've put forward a model, and we're working with people about an origin of life on land, where you have that power of wet-dry cycles that can cause organic monomers to polymerize, becoming longer, more complex molecules heading towards RNA and DNA. And we're very excited by that prospect. And so we've been doing a lot of research now on hot springs, active hot springs from New Zealand with colleagues out of the University of Auckland. And we're applying that to the search for life on Mars. And there's a little picture in front of you in the mid-right, the reddish kind of picture with these bumpy white things. Those are deposits from a hot spring on Mars that have already been visited in the past by the Spirit rover. And we think those are incredibly exciting targets to search for evidence of life, because all hot spring deposits on Earth, going back to 3.5 billion years ago, retain signatures of life. And so we're actually trying to develop a mission with the Japanese Space Agency and colleagues in Japan to go to Mars, bring those samples back, and analyze them here for signs of life. So that's a little bit of our program. And thanks, Anna, for the opportunity, and I'll hand it back to you.
Okay, thank you so much, Martin. And yeah, thanks for giving that great overview. So you might notice that the ACA is also very big on public outreach and science communication. So quite a few of our members are celebrities. Like Martin's a bit of a celebrity if you Google him. Another one of our celebrities is Pali. So he is a supramolecular chemist at the School of Chemistry with Albert and myself. And Pali has a lot of expertise working on peptide RNA interactions, liquid-liquid phase separations, hydrogels, and polymersomes, and has a huge interest in working on origins of life in the context of understanding it as a systems chemistry problem. And he also has some, he's done a lot of public outreach, and you can look up the public service announcements for why soap works in killing viruses. Martin already talked about Brendan's work. We have other biologists on board as well. So Belinda Ferrari is an associate professor that spends a lot of her time in Antarctica, actually. And so the idea is to look at extreme terrestrial ecosystems, looking for signs of life, identifying what life is there, and also trying to figure out what the survival strategies for these microorganisms are. And one of the big sort of projects that they've been working on is actually uncovering the presence of microorganisms that can basically live off air. So that was quite big news. It was published in Nature. So definitely look into that as well. And just a little plug for our astrochemistry colleagues. So Martin did mention that we have astrophysicists that are interested in the search for life in the universe, but we also have astrochemists. A lot of them are spectroscopists. So we have Chris Medcraft, Tim Schmidt, Laura, Chris Hansen, and Scott, who are all very much interested in astrochemistry, ranging from theoretical to experimental expertise. So lots of names for you to look up, but yeah, we have a lot of people who are passionate about astrobiology and related sciences at UNSW. So please, yeah, give these people a Google. So I wanted to talk a little bit about my recent work, and this is not trying to make molecules as such, but looking at the self-assembly of molecules into membranes. And so a bit about my background. I'm in the School of Chemistry now, but I didn't do it in undergrad. So I don't know why I'm saying this on the record, but I did everything except for chemistry and geology. And now I'm trying to dabble in chemistry and geology. So my senior thesis project was actually on optical fibers, and that was a lot of fun. Got to make some artwork. And then I went to Harvard to do my PhD. From, going from Sydney to that place was quite an experience, but I actually had a really good time. I even enjoyed the winters. My supervisor, Vinny, ran a lab that's very much about doing science slowly and carefully, and one of the main tools we used was digital holographic microscopy. So I'm a huge fan of being able to see things under the microscope, and the joy of being able to see E. coli do its tumble in 3D in real time is just fantastic. So I've always stuck with microscopy as a tool since then. And I worked on projects like understanding how solid colloidal particles, so like little plastic balls, self-assemble at an oil-water interface. And believe it or not, there was pretty rich physics in that. So I'm pretty into looking at a question very, very deeply, even if the question itself is quite simple. I was absolutely astonished to go to a talk by Professor Jack Shostak. And, you know, I didn't really think that I would understand anything, but then I was captivated by the idea of understanding the origins of life from a self-assembly perspective. Like if you have a bunch of molecules, how do they create something greater than the sum of the parts without necessarily rearranging the bonds? And the fact that molecules could rearrange into something that you could see under a microscope, that's what got me. So then I applied for a fellowship from the NASA postdoctoral program, and I got it. So that really helped me sort of change fields and start working on astrobiology. Oh, this picture is what Boston looks like the other half of the year. It's pretty nice. So I crossed the river to start working at MGH with Jack. So since then, Albert's mentioned that, you know, we work in this great building at UNSW. This was what the architects promised, and this is what we got. It's pretty similar, actually. It's very nice. And this is what part of the lab looks like now. In addition to all the equipment that Albert mentioned, I'm a microscopist, so sort of gathering capabilities there. And yeah, please contact me if you're curious about what we're trying to build and what measurements we're trying to make. This is my favorite part of the whole lab. This is in Albert's gas lab, where it is the origins of life control panel, and Albert has the power to hit the reset button on all of life. So yeah, don't mess around with Albert because he has the reset button. Anyway, so yeah, again, I'm a microscopist, so I'm interested in working on things that I can see, and we can see cells under the microscope, and it's really that is the sort of richness of life around us. So even if we think about food, we don't really think about what happens at the cellular level, but microorganisms give us all these delicious fermented foods. Basically every culture in the world has discovered the delights of fermentation. And we also have not just unicellular organisms, but multicellular organisms. So huge variations on this one theme of a cell, which is this fundamental unit of life. And what delineates a cell is the cell membrane. And so cells do at the unicellular level all these processes, like they consume nutrients and let waste out. Cells also grow and divide in order to propagate, and they can also fuse with other cells or cell-like objects to transfer chemicals and information. So pretty cartoon level explanation of what cells are. And the part I'm really wanting to talk about today is the cell membrane itself, that delineating boundary. And so in biology textbooks, you'll often see this like super complicated thing, which cells definitely are. They're way more complicated than that cartoon. And the envelope on the outside actually consists of what we call a lipid bilayer. So it's a bunch of phospholipids. We call them phospholipids because they have a lipid tail and they have a phosphate-based head group. And because of the hydrophobic effect, they arrange themselves into a bilayer. So that it's like this thin lipid envelope that sort of encapsulates everything and creates a barrier. And what I wanted to impress you with was the idea that these things are really thin, but they're really tough. So if I show you that house that I actually spent a lot of time in writing my PhD thesis, if we blew up the cell to the size of that house in size, the thickness of the cell membrane would be about as thick as an iPhone. And this thing is just, it's so thin, but it kind of just holds everything in. So it's quite remarkable from a material science perspective. And it does all these other things as well, like it's responsible for transport, and it also needs to divide for the cell in order to divide the cell. What I haven't shown you in these cartoons is that very thin wall actually doesn't do much by itself. What does all the work are the proteins embedded inside the membrane. Membrane proteins are typically 40% by weight in the membranes. And they do all of those interesting tasks. And even making the phospholipids themselves requires a bunch of membrane proteins. So if we remove all the proteins that make the phospholipids and even divide the cell, what you're left with is an inert sac, this 5 nanometer thick, very tough membrane, but it's very inert, it's very stable. All of the exciting processes are actually conferred by proteins. So a lot of what I'm trying to do in lab now, and what I started in Jack's lab, is actually to think about what can we accomplish without proteins? How can we do all these processes in as simply as possible? So I mentioned earlier that phospholipids rearrange into a bilayer in water, and that's a self-assembly property of these sorts of molecules. And the property of the molecule that makes it do this is that it's amphiphilic. There's a hydrophilic side and a hydrophobic side. And something else that we encounter on a daily basis that's hydrophilic and hydrophobic is soap. So soap molecules are also amphiphilic molecules. We all know that if you give a soap molecule an oil-water interface to go to, for example, some oil that's on your hands and you're running water, the soap will coat the oil droplets and help you remove them. But if you don't have any oil at all, it turns out that they can actually form bilayer membranes. And this was reported actually by Australian scientists in the 1970s, Gibicki and Hicks. They were at Macquarie University, also in Sydney. And basically said under the right conditions, you can get these fatty acids or soap, which are subunits of phospholipids, to self-assemble into a membrane. And this is pretty exciting because when people have analyzed the organic material on meteorites, they found some simple fatty acids. So what this is saying is not that necessarily all the fatty acids on Earth came from meteorites, maybe some of them did. What it's actually saying, and why it's really exciting, is that we don't need biology in order to make fatty acids. They're actually simple enough to just be made in space. And the other thing is they're kind of a subunit of the modern cell membrane lipids. So that's kind of nice. Cool. So we know fatty acids as soap. When they're at a high pH and they're deprotonated, they form micelles and they work really well as a detergent or soap. If you drop the pH a bit, what you end up getting are these bilayer membranes. And I'll just show you what they look like. Basically, if you dump any membrane forming molecule into water or buffer, they end up self-assembling into these compartments that can encapsulate material. So we can have some RNA that's conjugated to a green dye, so it looks green under a fluorescence microscope, and we can label the membranes red. And what you end up seeing is these are pockets of green that are trapped inside the membranes. And when I saw this, as a physicist, I just really, really wanted them to all have one single layer, because that makes them much easier to study, but it also makes them look more like cells. Like if you dye the cell membrane of a yeast cell, they don't look like onions necessarily. There's just one nice cell membrane on the outside, which biologists frequently call the plasma membrane. So I was seeing these onions and I wanted to make single layered ones. And very much like what Albert was saying, one of the aims in prebiotic chemistry is just to interfere as little as possible as humans, right? We don't want to go in and do the self-assembly for the system. We just want it to do it itself. And so during my postdoc, I tried methods like microfluidics and other techniques, but what I really wanted to do was avoid human intervention and still make these single layered fatty acid vesicles. And that's what I kind of discovered by accident was a way to make these. And so you can read about this more in a paper that we published very recently. Basically, there's a self-assembly method to get fatty acids or soap molecules to rearrange into these single layered vesicles. And so I now have a model system to work with that doesn't really require human intervention. I have to mix the ingredients in the beginning and then leave it overnight, just agitating. That's pretty much it. And what we found is that even though these membranes are very thin and they do self-assemble, you can trap all sorts of things inside. So we can trap dye inside, we can trap nanoparticles inside, and we can even trap colloidal particles inside. So this is just polystyrene, but we've made these vesicles in the presence of marillinite clay, and clay particles can end up trapped inside. So these are pretty good vessels in terms of like a little micro-reacting compartment. So finally, I'll just say, we want to be able to do things to these membranes now without using any proteins. And so one of the first things that we tried doing was seeing if we could get them to divide. So we get them to grow just by feeding the membrane with extra membrane material. So we add some soap to some vesicles and see what happens. And you'll see that the surface area increases. That makes the membranes a bit wobbly. But then what happens afterwards is that these wobbly membranes pinch off into spheres. And again, I have further details in the paper, so feel free to ask me questions or look at the paper itself. But what we found is the reason why these membranes are able to pinch off into spheres and why they like being spheres is probably because these membranes are surprisingly rigid. And when you have a rigid membrane, it means that they can store elastic energy. So if you increase the surface area to volume ratio and you create stresses in the membrane, the elastic stress can be relieved by pinching off into smaller spheres. And so what is normally a very energy intensive process in a cell, we actually kind of almost get for free in our system just by virtue of having a very dynamic yet very stiff rigid membrane that can store elastic energy. So great, so this is the big picture of what we're trying to build in my lab right now. We have students working on all sorts of these problems, and they're all really hard problems, so any sort of collaborations or feedback is more than welcome. And I'd like to thank my theory collaborator, James Kint from Emory, and also Jack from Mass General, and my funding for this particular project